Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. I am EndangWiniati Bachtiar from Faculty of Dentistry, Universitas Indonesia. Let me talk about our ongoing project, and the title is Does microRNA in salivary exosome reflect the state of COVID-19 infection? Our team consists the lecturer and the doctor from the Department of Oral Biology and Oral Science Research Center, Faculty of Dentistry, Universitas Indonesia, Department of Periodontology, Faculty of Dentistry, Universitas Indonesia, and a colleague from the Universitas Indonesia General Hospital, or RSUI, Depok, Indonesia. Okay, the introduction. A microRNA is a single-stranded RNA found in various biological samples, including saliva. These molecules regulate gene expression at the post-transcriptional level. miRNA have been studied as a biological marker to help establish the disease's diagnosis, severity, and prognosis. In the case of COVID-19 infection, oral cavity plays as a reservoir of SARS-CoV-2 virus. As we know, angiotensin-converting enzyme 2 or ACE2 is the primary receptor expressed on the host cell for SARS-CoV-2 infection. And this molecule also plays an essential role in the entry of SARS-CoV-2 into cell to subsequently cause late infection COVID-19. Oral mucosal tissue that express ACE2, especially in tongue epithelial cell, are organisms that can act as intermediates or reservoir potential for COVID-19 transmission. The potential use of the saliva as a diagnostic from the transmission of SARS-CoV-2 has been reported by Zhu et al. In 2019. Here we can see that the host cell of the oral cavity, such as the epithelial, will express the ACE2 molecule as a receptor binding of SARS-CoV-2 virus, and as well as we can find the SARS-CoV-2 receptor in the salivary gland and the other side of the oral mucosal niche. Hence, it is not surprising that the aerosol or droplet from the oral cavity can be as a source of SARS-CoV-2 transmission in the population. And the saliva is a biological fluid that can be used as an alternative to the diagnosis of viral infection because it is easy and quickly collected without the use of special tool and reduce the discomfort of patient during the sample collection. The angiotensin-converting enzyme 2 and furin protein expressed in various grades in the different oral anatomical sites. Here we can see that furin, the most strongly positive expression in the lip, and then a mild positive is furin in the gingival tissue, and then ACE2 in buccal and lip mucosa, as well as in the tongue. Here, ACE and furin is positive in mild expression, and then very weakly expressed in the palatal of the oral mucosa. Salivary exosome that consists of microRNA have been reported can be used for diagnostic profiling from both sources, as well as provide a new insight into the molecular mechanism involved in the disease progress. The use of saliva in virus detection and detailed the comprehensive literature describing the systemic correlation of saliva with a viral activity. So, salivary exosome microRNA could be reflect the state of COVID-19 infection. So our question is, how come the microRNA will be used as a genetic marker to reflect the state of COVID-19 patients? Here we can see that during the infection of SARS-CoV-2 to the epithelial cells or the host cells, the virus will use the genetic material from the host for the propagation or proliferation of the virus. Hence, the microRNA will be expressed both genetic materials and the SARS-CoV-2 genetic materials. Yes. Lastly, we can see that the microRNAs will be express both genetic material from both virus and the host cells. Come to the conclusion. For future direction, ongoing research to investigate the relationship between the presence of microRNA isolated from the salivary exosome with IgG and IgM status of anti-COVID SARS-CoV-2 patients and the correlation with the CT value of RT-PCR of SARS-CoV-2 will be evaluated. And we propose that the salivary exosome will reflect the state of severity of COVID-19 patient. Lastly, let us acknowledge the author would like to express the appreciation for the support of Ristek BRIN with the project number NKB 140. Thank you very much. Thank you very much for the opportunity to share of our ongoing projects and hear a little bit about the take-home message. Please follow the science to cope the problem of the pandemic of COVID-19. Thank you very much.